A ranch dog named Shep dropped a small masked animal at Lucille Hogg's Wyoming door in September 1981. Biologists had declared the black-footed ferret functionally extinct 2 years earlier, but that dead animal in the dirt meant something impossible was true. Somewhere beneath the Wyoming grassland, a ghost population was still breathing. Does someone go from a single dead animal on a doorstep to the most consequential wildlife recovery in North American history? The black-footed ferret is built for one thing: the underground. Slender, nocturnal, masked like a bandit, engineered to slide through prairie dog tunnels at full speed, navigating total darkness by instinct and smell. The ferret doesn't visit prairie dog burrow systems. It moves in permanently. Approximately 90% of its entire life happens underground, and roughly 90% of its diet is prairie dog. A single adult needs between 100 and 150 prairie dogs per year just to survive. No other large North American predator is locked this tightly to a single prey species inside a single underground architecture. Remove the prairie dogs and you haven't degraded the ferret's habitat. You've deleted it entirely, and that wasn't even the part that got people angry. Ecologists celebrating the prairie dog's resilience in the mid-20th century missed what was already collapsing beneath the surface. Someone saw it coming. Ecologists studying prairie dog colony dynamics noted that disappearing predators were already destabilizing colony behavior. Prairie dogs expanding unchecked in some areas, crashing in others. Nobody listened. The collapse did not happen overnight. It happened the way most ecological disasters happen, incrementally, through 100 separate decisions made by people who never once considered what lived beneath the ground they were reshaping. Through the 19th and into the 20th century, the Great Plains underwent one of the most dramatic land transformations in human history. Settlers moved west, broke sod, planted crops, and grazed livestock across a landscape that had evolved over millions of years. Prairie dogs were not seen as part of that system. They were seen as competitors. Their burrows were hazards for livestock. Their colonies consumed grass that ranchers wanted for cattle, and so the campaigns began. Poisoning operations targeted prairie dog towns with systematic precision. The United States government ran coordinated eradication programs that blanketed millions of acres with poison grain, strychnine, and later compound 1080. By some estimates, prairie dog populations were reduced by 98% from their historical range during the 20th century. Colonies that once stretched for hundreds of continuous miles were reduced to isolated fragments, separated by plowed fields and poisoned ground. For the black-footed ferret, each poisoned prairie dog town was a neighborhood erased. The ferret cannot cross open agricultural land. It cannot survive without active burrow systems to shelter in. So as the colonies shrank and fragmented, the ferret population shrank in exact parallel. The two species were bound together in decline, the same way they had been bound together in function. Then came the second blow, sylvatic plague caused by Yersinia pestis. The same pathogen responsible for the Black Death in medieval Europe arrived in North America from Asia in the early 20th century. Prairie dogs have no evolutionary history with this pathogen. They developed no immunity, no behavioral resistance. When plague entered a prairie dog town, it moved through the colony like fire through dry grass. Mortality rates in affected colonies could reach 100% within weeks. The plague did not need human help to spread. It moved through flea populations across the landscape, jumping from colony to colony. And because the ferret hunted prairie dogs and lived inside their burrows, it was exposed to plague-carrying fleas constantly. By the 1970s, confirmed ferret sightings had essentially ceased across the entire range. A formal survey conducted in 1979 declared the species likely extinct. Not endangered, not critically depleted, extinct. The scientific consensus was that the black-footed ferret had already crossed the threshold from which no return was possible. Ecologists celebrated what they thought was a contained problem. They shouldn't have. What the survey could not account for was what was happening to the prairie itself in the ferret's absence. Without burrowing disturbance, soil compaction increased. Compacted soil sheds rainfall rather than absorbing it. Water runs off the surface, reducing moisture available to plant roots and accelerating erosion. Plant diversity dropped as disturbance-dependent species lost their footholds. The grassland was becoming a simpler, less resilient system with each passing decade. The ferret's disappearance was not just the loss of a single species. It was a symptom of a systemic underground collapse, a failure of the subterranean architecture that had shaped the Great Plains for thousands of years. Picture a Wyoming ranch in September 1981. The light is flat and cold. A dog named Shep drops something at the door, something small, masked, and dead. Lucille Hogg does not know what she is looking at. She takes it to a local taxidermist, who does not know either. It eventually reaches a biologist, and the biologist goes very still. It is a black-footed ferret, a species declared likely extinct 2 years earlier, a species that, according to the best scientific consensus of the time, no longer existed on Earth. But here it is, unmistakably real, unmistakably dead, and unmistakably proof that somewhere in the grasslands around Meeteetse, there are more. Field surveys mobilized quickly. Biologists combed the prairie dog colonies surrounding the hog ranch, scanning at night with spotlights. Ferret eyes reflect a distinctive green shine in artificial light, and they found them, a surviving colony hidden in the shortgrass prairie of the Bighorn Basin, persisting in a landscape that had somehow escaped the worst of the poisoning campaigns. At its peak, the Meeteetse population reached approximately 130 individuals. For species believed extinct, this was extraordinary, but the genetic analysis told a more cautious story. The population was thin, isolated, and carrying the accumulated vulnerability of years of inbreeding. The scientific community was cautious about intervention. The ferrets were alive and reproducing. The argument for leaving them alone was real, and it had weight, but the ferret wasn't done yet, not even close. In 1985, 2 disasters arrived simultaneously. Sylvatic plague swept through the Meeteetse prairie dog colonies, collapsing the prey base, and canine distemper, a viral disease to which black-footed ferrets have essentially no natural resistance, moved through the ferret population itself. Within months, the population that had peaked at 130 animals crashed to approximately 18 confirmed individuals. 18, the entire species compressed into 18 living animals in a single location in Wyoming. The debate that followed was one of the most consequential in the history of American conservation. Leave the remaining ferrets in the wild, where plague and distemper remained active threats, or capture every last individual and attempt to build a captive breeding program from scratch, with no guarantee that captive breeding would work, no guarantee that the animals could survive capture and captivity, and no guarantee that anything bred in captivity could ever be returned to the wild. Wyoming Game and Fish Department and the United States Fish and Wildlife Service made the call. Capture them all, every single one. The risk of losing the species entirely in the wild was judged to be greater than the risk of the captive program failing. It was an all-or-nothing decision made under conditions of radical uncertainty, with the entire existence of a species hanging on the outcome. The last wild ferret was captured in 1987. For the first time in its evolutionary history, the black-footed ferret existed only in cages. The rooms were small and carefully controlled, temperature monitored, lighting on a precise schedule. Every animal cataloged, tracked, and assessed for health with a thoroughness that bordered on obsessive, because obsessive was exactly what the situation demanded. 6 facilities across the United States were established as insurance nodes in a distributed network. The logic was cold and clear. No single disease outbreak, no single facility failure could be allowed to wipe the entire captive population. Distribution was survival, but distribution alone could not solve the deeper problem. All 18 of the Meeteetse survivors had been captured, but not all of them would contribute to the breeding program. By the time the genetic accounting was done, only 7 of the original 18 individuals successfully produced offspring that survived and reproduced. 7 founders. The entire genetic future of the black-footed ferret would descend from 7 individuals. The implications of a 7-founder bottleneck are severe, and they compound with every generation. Inbreeding depression begins to manifest quickly. Immune function narrows. Reproductive rates decline. Developmental abnormalities appear. Every pairing decision made by the breeding program was not just a matter of producing offspring. It was a genetic calculation, an attempt to maximize the diversity that remained while preventing the most damaging inbreeding combinations. The behavioral dimension added another layer of complexity. Ferrets raised in captivity, fed pre-killed prey, and never exposed to the threats of the open prairie were not automatically equipped to survive reintroduction. They had to learn to hunt live prairie dogs. They had to learn to recognize and evade predators, golden eagles, badgers, coyotes that they had never encountered. Behavioral conditioning programs were developed, covering live prey training, predator exposure protocols, and burrow habituation exercises. Some animals adapted, others did not. The early years were brutal. Disease moved through captive populations despite the precautions. Pregnancies failed. kits died. There were moments when the program appeared to be failing faster than it was succeeding. If the program had stopped here, this would be a very different story, but the losses were absorbed, the protocols were revised, and the numbers slowly, painfully began to move in the right direction. By the early 1990s, the captive population had grown to the point where the first experimental reintroductions became possible. The ferret was ready to go back. The question was whether the prairie was ready to receive it. Shirley Basin, Wyoming, 1991. Biologists opened transport carriers on a cold autumn night, and black-footed ferrets step out onto prairie dog ground for the first time in years. The animals pause at the burrow entrances, testing the air. Then they go down. The first reintroduction of the black-footed ferret had begun. The early results were sobering. Survival rates among reintroduced animals were low. The captive-raised ferrets were entering a landscape that had changed significantly since the species disappeared. Prairie dog colony densities were different. Predator communities had reorganized in the ferret's absence. And plague was still out there. Yersinia pestis had not gone anywhere, and without active management, it remained fully capable of collapsing a prairie dog colony within weeks. The plague problem proved to be the dominant constraint on recovery for the next 2 decades. Reintroduced ferret populations that showed early promise would stabilize, begin to reproduce, and then collapse as plague swept through the prairie dog colonies beneath them. Site after site demonstrated the same pattern: establishment followed by growth followed by crash. Without plague management, reintroduction was essentially a slow-motion exercise in futility. Researchers at Kanata Basin in South Dakota watched a population they had carefully built over several seasons get cut in half by a single plague event in 2008. The animals were there. The habitat was there. The plague didn't care. The management response evolved. Dusting programs using insecticide-treated dust pushed into burrow entrances began to suppress flea populations, the primary vector carrying Yersinia pestis between prairie dogs. Oral plague vaccines, delivered in peanut butter-flavored baits, distributed across prairie dog towns, were developed and field-tested by United States Geological Survey researchers starting in the mid-2000s. These interventions were labor-intensive and expensive, requiring annual treatment of thousands of acres across multiple states. But they worked. Sites with active plague management showed dramatically better ferret survival and reproduction than unmanaged sites. At some managed locations, annual ferret kit production doubled compared to unmanaged controls measured in the same years. Where ferrets did establish, something measurable began to happen in the prairie dog colonies beneath them. Burrowing intensity increased. Prairie dog behavior shifted. Colonies restructured spatially in response to predation pressure from below. Vegetation patterns around active prairie dog towns began to change as the disturbance regime that ferret predation helped regulate came back online. The prairie was responding, slowly, unevenly, but measurably. Reintroduction had exposed something captive breeding could not have predicted. The ferret was not returning to an unchanged ecosystem. It was returning to a system altered by its absence, and its return was itself an alteration, a reintroduction of a regulatory signal the underground architecture had been missing for decades. The prairie was different, and so in ways nobody had fully anticipated was the ferret's role within it. Pull back the grass and look at what is actually happening beneath the surface of an active prairie dog town, and you are looking at one of the most productive and ecologically complex microenvironments in the entire grassland system. The excavation never stops. Prairie dogs are constantly digging, extending tunnels, opening new chambers, pushing subsoil to the surface in fresh mounds that ring the burrow entrances. This constant turnover brings mineral-rich material from deep layers up into the root zone, creating microsites of unusual soil chemistry that seed rare plant species unable to establish in the surrounding compacted grassland. The ferret's role in this process is not direct. Ferrets do not dig the mounds. But ferret predation pressure is what regulates the size and spatial structure of prairie dog colonies, preventing any single area from being overgrazed while maintaining the colony density necessary for burrowing disturbance to continue. Without predation pressure, prairie dog colonies either collapse under plague or expand into dense clusters that strip vegetation from large areas and then crash. The ferret is the regulator that keeps the system in the productive middle range. The hydrological function of active burrow systems is striking. Compacted prairie sheds rainfall from its surface. Active prairie dog towns, with their network of tunnels functioning as vertical infiltration channels, absorb significantly more rainfall per unit area. In dryland systems where annual precipitation can fall below 12 inches, the difference between shedding and absorbing that rainfall is not a marginal ecological footnote. It is the difference between a functioning grassland and a degrading one. Soil microbial communities tell a similar story. Researchers comparing soil samples from inside active prairie dog towns against collapsed colony sites have found measurable differences in nitrogen cycling rates, fungal network density, and decomposition activity. The underground biological community is more diverse and more active inside functioning prairie dog infrastructure than outside it. Consider what that means at scale. At least 136 other species depend on prairie dog infrastructure that the ferret helps sustain. Burrowing owls nest in abandoned burrow chambers. Swift foxes shelter in the same tunnel networks. Mountain plovers preferentially nest in the short-cropped vegetation around active prairie dog towns. Ferruginous hawks concentrate their hunting over colonies where prey density is highest. These species do not depend on the ferret directly. They depend on the prairie dog system that the ferret helps sustain. Remove the ferret, and the regulatory pressure that keeps that system functioning disappears. The cascade runs upward through the soil and outward across the landscape in ways that connect species that appear to have nothing to do with each other. The underground engine is not a metaphor. It is a measurable, documented, ecologically critical system, and the black-footed ferret is the key that keeps it running. Every black-footed ferret alive today carries a genetic signature that traces back to the same 7 animals. 7, not 17, not 70. 7 individuals whose reproductive success in the early years of the captive program determined the entire genetic architecture of the species going forward. The other 11 Meeteetse survivors either failed to reproduce in captivity or produced offspring that did not survive. Their genetics, whatever unique immune variants, whatever disease resistance pathways they carried, died with them. The consequences of a 7-founder bottleneck accumulate silently across generations. Inbreeding depression does not announce itself with a sudden catastrophic failure. It erodes fitness gradually, immune function narrows incrementally, reproductive rates decline by degrees, developmental abnormalities appear with increasing frequency. By the 2000s, researchers tracking captive and reintroduced populations were seeing the signs: reduced litter sizes, elevated rates of pregnancy failure, immune profiles showing concerning uniformity across individuals that were nominally unrelated but genetically nearly identical. The disease resistance problem was the most alarming dimension. A population with high genetic uniformity in immune pathway genes is a population with a single immunological profile, and a single immunological profile is a single point of failure. A plague strain that evolved to exploit the specific immune architecture shared by all living black-footed ferrets could, in principle, move through the entire species the way the original plague swept through prairie dog towns, completely and without survivors. Scientists working on the recovery program began searching for a way around the bottleneck. The answer came from an unexpected direction: the past. Tissue samples collected from ferrets that had died before the captive breeding program began. Animals from the Meeteetse population that had not survived long enough to contribute to the breeding effort had been preserved in frozen storage at the San Diego Zoo's Frozen Zoo Biobank. These samples contained genetic material from individuals outside the 7-founder lineage. Genetic material that had never entered the living population. Immune diversity, disease resistance pathways, and developmental variation that had been locked in a freezer for decades while the living population narrowed around its 7-founder core. The question that emerged was one that no conservation program had ever seriously had to confront before. Could genetic material from a ferret dead for 30 years be used to produce a living animal? And if it could that animal's genetics be bred into the living population in a way that meaningfully broadened the species' immune base? The answer to both questions would turn out to be yes. But the path to that answer was longer and more complicated than anyone anticipated. In December 2020, Revive and Restore and the United States Fish and Wildlife Service announced that a black-footed ferret had been successfully cloned from frozen tissue samples collected 30 years earlier. The cloned ferret was named Elizabeth Ann. She was alive, healthy, and genetically unlike any living ferret on Earth. Her donor was a ferret named Willa, who had died in 1988, before the captive breeding program had fully established itself, before anyone understood the genetic ceiling that the 7-founders would impose. Willa had never bred in captivity. Her genetics had never entered the living population. She had been, from the perspective of the recovery program, a genetic dead end. But her tissue had been preserved in the San Diego Frozen Zoo, and 30 years later, her cells were still viable enough to provide the nuclear material for a cloning procedure. A dead ferret's 30 30-year-old frozen skin cell became the genetic blueprint for a living animal. The technique used was somatic cell nuclear transfer, the same fundamental approach used to clone Dolly the sheep in 1996. A skin cell from Willa's preserved tissue provided the nucleus, transferred into an enucleated domestic ferret egg cell. The resulting embryo was implanted into a surrogate. Elizabeth Ann was born on December 10, 2020. What Elizabeth Ann carries is not simply a curiosity. Her genome contains genetic variants absent from every single living black-footed ferret. Immune pathway genes the 7 founder population never had. Disease resistance variants that disappeared from the living gene pool when Willa died without offspring. These variants represent the difference between a species with one immunological profile and a species with multiple, the difference between a single point of failure and a more resilient genetic architecture. The goal was never to produce one cloned ferret and call it a success. The goal was to breed Elizabeth Ann's lineage into the wild population, to use her offspring as vehicles for introducing Willa's genetics into the living gene pool at a scale that would meaningfully shift the species' immune diversity. This is genetic rescue, not replacing the existing population, but augmenting it with diversity that was lost and has now been recovered. Elizabeth Ann's successful birth marked the first time cloning had been used for the conservation of a United States endangered species. Tissue samples from dozens of endangered species are preserved in biobanks around the world. Genetic material from animals that died before conservation programs existed. Elizabeth Ann demonstrated that this material is not merely archival, it is potentially functional. It can be brought back into living populations. That possibility, cloning from preserved tissue as a standard tool in conservation genetics, has permanently expanded what conservation can attempt. Where ferrets have returned to the prairie, the evidence is accumulating in the soil itself. Sites with established ferret populations show measurably higher plant species richness in and around active prairie dog towns compared to ferret-absent control sites surveyed in the same years. The difference is not subtle at the microscale. Burrow mound microsites support plant assemblages that simply do not appear in the surrounding compressed grassland, disturbance-dependent species that require the churned, mineral-exposed soil that only active burrowing creates. Water infiltration rates tell a parallel story. In dryland systems across shortgrass and mixed grass prairie, functioning burrow complexes absorb rainfall at rates measurably higher than the surrounding compacted ground. In regions where annual precipitation can fall below 12 inches, that infiltration differential is not a footnote. It is the mechanism by which the grassland retains enough soil moisture to sustain deep-rooted plant communities through dry seasons. The difference between a prairie that holds its water and one that sheds it is the difference between a system that can recover from drought and one that cannot. Grassland bird communities are tracking the recovery. At reintroduction sites where ferret populations have stabilized, burrowing owl densities have increased as abandoned ferret-modified burrows become available for nesting. Mountain plover, a species that preferentially nests in short crop vegetation around active prairie dog towns, has shown population rebounds at several monitored sites in Wyoming and South Dakota. Swift fox, which uses prairie dog burrow infrastructure for denning, has recolonized areas where prairie dog colony health has improved under ferret predation pressure. The carbon dynamics are less visible but measurable, as deeper-rooted plant communities re-establish in the disturbed microsites around active burrow systems. The soil organic matter profile begins to shift. Deeper roots deposit carbon further down the soil profile, where it is less vulnerable to surface oxidation. The grassland is not simply recovering its species composition, it is recovering its capacity to function as a carbon-storing system, slowly, incrementally, but in a direction that decades of degradation had reversed. The ferret has become a measurable proxy for prairie ecosystem function. Where ferrets persist, the underground engine is running. Where it is absent, the metrics converge on the same pattern: compressed soil, reduced infiltration, narrowed plant diversity, declining bird communities. The animal is small enough to hold in 2 hands. The system it regulates spans millions of acres. When you put this all together, what you are looking at is not a single species recovery. It is a demonstration that restoring one underground predator can reboot an entire ecosystem. Over 300 wild ferrets now range across more than 30 reintroduction sites in 8 US states, Canada, and Mexico, descended from 18 animals and 7 genetic founders. The genetic ceiling imposed by that bottleneck is no longer fixed. Elizabeth Ann's lineage represents a door that was thought permanently closed, now open. The lessons extend well beyond the ferret. Degraded rangelands, dryland watersheds, and grassland restoration projects worldwide share the same underground logic. The surface is shaped by what happens below it. The burrowing, the disturbance, the infiltration, the microbial cycling. These are not secondary processes. They are the engine, and the engine needs a regulator. The Great Plains underground was never silent. It was waiting for the masked predator to come back and start it again. If this kind of story is what you come here for, subscribing keeps more of it coming.